Very good morning to one and all. This is Mrs. Mary Ratnanita once again welcoming you back to your pharmaceutical microbiology class. The topic for today's R class is structure of antigen and antibody. So we have already learnt about the antigen, like the different types of immunology. We know now we know what is immunology, what is immunity, and what are the different types of immunity which our body carries on. So in while we were studying about immunity and immunology, we came across these two terms many a times, antigen and antibody. So let us see what are these antigens and antibodies, and what is their structure and how are they important, and what is like how they contribute to the field of immunology. We'll see in this topic. Now, talking about before entering into the topic, let us learn about the learning objectives. So the student shall be able to define what is antigen, and the student should be able to, also able to explain the structure of antigen, also the structure of antibodies and how the antigen binds to antibodies, and how these antigen, what are the different properties of these antigens and antibodies, and how are they involved in immunity in our immune system. All will be dealt in this topic, and the student will be learning all these topics by the end of this topic. Now, coming to your main topic, antigen. As we have already learned about what is antigen in our previous class, we have already seen the definition part. Let us remind it refresh it once again. So, antigen, what is an antigen? We have already learned that it is macromolecular entity that essentially elicits or stimulates an immune response how by production of specific antibodies in the body of the host. So, what are we saying? These are the macromolecules, and these have the capability of stimulating the production of antibodies where in the body of the host. And most of the antigens are protein in nature, but some can be like carbohydrates or nucleic acids or lipids, etc. But mostly, almost all the antigens are, most of the antigens are proteins in nature. And when you talk about the types of uh, antigen, so these antigens are of two types, that is complete antigens and incomplete antigens, which are also called as haptens. We have already learned about the definition of haptens also. We'll see what is this as we proceed further. So when we talk about complete antigens, so these are the antigens which are able to induce antibody formation by themselves. That means these antigens has the capability of producing antibodies by themselves without anybody's help, and produce a specific reaction with that particular antibody. So whatever the antibody has been produced by the stimulation of this particular antigen, that antigen will react with that particular antibody specifically. So that is nothing but your complete antigens. And generally these complete antigens are high molecular weight proteins, but some are polysaccharides in nature. Whereas when you talk about incomplete antigens or hapten, we have already seen definition of hapten in our previous class. So what are these haptens? These are chemical. These haptens are nothing but these are also chemical substances of low molecular weight, and these haptens have no capability of either stimulating the immune response or producing antibodies. But when these haptens are linked to proteins called as carrier proteins, either in vitro or in vivo, they can react specifically with its corresponding antibody. So what are we saying? This basically these haptens does not have the capability of inducing the immune response or producing antibodies. But when these haptens are combined with carrier proteins, either in vitro or in vivo, these haptens gets the capability or the power to react specifically with its corresponding antibody. So these are nothing but your haptens. So these are incomplete antigens. What are complete antigens? Complete antigens are nothing but these are the antigens that has the capability of producing, stimulating the immune response and producing antibodies and reacting specifically with that particular antibody. So these are true antigens. When you talk about incomplete antigens or haptens, incomplete antigens or haptens are nothing but these haptens are chemical substances with low molecular weight, and these does not have any capability or capacity to induce stimulation, immune response, or produce antibodies. But when they are combined with carrier proteins, they have the capability of uh, they have the capability to react with a specific antibody. Now, generally, the carrier proteins, what these haptens are combined are generally serum proteins like your albumins, globulins, or synthetic polypeptides. So these are the examples of the carrier proteins with which the haptens will be combined. And when you talk about the types of haptens, there are again two types of haptens: simple hapten and complex hapten. When you talk about simple haptens are nothing but low molecular weight chemical substances, and which these low molecular weight chemical substances, when combined with specific antibody, do not form any precipitate. So simple haptens are low molecular weight chemical substances, and these chemical substances, when they are combined with an antibody, they will not produce any precipitate. Whereas when you talk about complex haptens, these are high molecular weight compounds which, when combined with an antibody, forms a visible precipitate. So that is the difference between your simple and complex haptens. Now, talking about the structure of antigens, what is antigen and how does it looks like? So you can see this is the general structure of antigen, and this is an antibody. So this antigen will go and fit in this area, and then it combines with antigen and antibody. And if at all this is complementary with this, then only it can bind with, antigen can bind with antibody. So a small area of chemical group on the antigen molecule that determines specific immune response and reacts specifically with antibody is called an epitope. So you can call this point as epitope. So this is nothing but it is a small area of chemical group which is present on the antigen. This is antigen, so this is present on the small area of chemical group which is present on the antigen molecule, and this specific area determines the immune response and it reacts specifically with the antibody, and that particular area is called as epitope. An antigen, the epitope is also called as antigen determinant site. So an antigen possesses several epitopes which differ in specificity. It's not that antigens has only one epitope, or that epitope only will fit into the antibody. No, antigens possess several epitopes which differ in specificity. For example, the number of epitopes in an antigen is referred to as its valency. So whatever the number of epitopes are present on the antigen, they are called as their valency. So these these epitopes are either functional or non-functional. Now, complete antigen has two or more than two functional valencies and some non-functional valencies. So as we said, you have complete antigens and incomplete antigens. So as we are saying that antigen is a chemical substance and it contains number of several epitopes, and these epitopes differ in their specificity, and the number of epitopes on an antigen is referred to as valency. So when you talk about the complete antigens, these complete antigens contains two functional, and as I said, the epitopes, some are functional and some are non-functional. So when you're talking about complete antigens contains a minimum of two functional epitopes and remaining can be non-functional. Whereas the total valencies is nothing but functional valency plus non-functional valency. That means whatever the number you have. So for example, if you take this as an antigen, you have 1, 2, 3, 4, 5, 6, 6 epitopes. So out of 6 epitopes, you can only 2 will be functional and the remaining 4 will be non-functional. So 2 functional plus 4 non-functional will give you complete number of valencies. So this is the basic structure of your antigen and antibody and how they react with each other. And now talking about the properties of antigen. Now antigen has different properties. For example, you have foreignness. Under foreignness, you have isospecificity, autospecificity, organ specificity, heterogenic or heterophile antigen, and you have different properties like size, chemical nature, susceptibility to tissue enzymes, antigenic specificity, species specificity. All these are nothing but your properties of antigen. So let us see what are these properties and how does it go one by one. So talking about the foreignness, the first property, foreignness. So immune system has the ability to distinguish between self and non-self antigens. As I told you when I was explaining your cell-mediated immunity, the T cells has the capacity of recognizing the self and non-self antigens. So as I similar, that is what the statement says here. So immune system, our body basic immune system has the ability to differentiate between self and non-self antigens. Only antigens which are foreign to the individual will be attacked, and only the one which are self antigens will not be attacked by your immune system. So that is about your foreignness. And coming to isospecificity, so isoantigens or alloantigens are antigens. Uh, um, I mean, like these are antigens which are found in some, but not in all members of a species. So isospecificity. So what are we saying? These are the antigens or alloantigens or isoantigens. So these are the antigens that are found in some but not in all members of a species. For example, human blood group antigens and RH antigens. So these are present in, I mean, like you have A group, B group, AB, O, RH positive. So all the human beings will not have the same kind of antigens. So they might differ. So they might not present. They may not be present in all, but they are present in some of the particular species of members of the species. So now talking about autospecificity. So autologous or self antigens are non-immunogenic, but under certain circumstances, like under lens protein or thyroglobulins, they act as autoantigens. That means the self antigens will act as foreign antigens. So in case of damage to the thyroid gland or damage to the testes, or in case of injuries to the lenses, so what happens? These self antigens, they undergo damage, and these self antigens pretend to be the foreign antigens, and they try to attack our body. So that is nothing but autospecificity. So you also have autoimmune disorders, right? So what happens as self antigens will not recognize us. So as a result, they try to act as foreign antigen and try to create problems in our body. So the same thing, autospecificity. So generally, these are body self antigens, but they are uh, due to some reasons, or due to, like, for example, like damage to thyroid gland or damage to lenses or damage to testes, these antigens will uh, mm, I mean, they will not able to recognize themselves as self antigens, but pretend as foreign antigen and cause problems to the body. So that is nothing but your autospecificity. Now, organ specificity. So these are confined to a particular organ or tissue. So these are antigens, as the name itself says, organ specificity. So that means they are confined to particular organ or a particular tissue. So organs like kidney or brain or lens proteins or thyroglobulin of different species, they share same type of antigens. So these are organ specificity antigens. Talking about your heterogenetic or heterophile antigen. So heterogenetic or heterophile antigen. So some antigens are similar in nature, present in different tissues of different biological species or classes or kingdom. So what are we saying here? These antigens are similar in nature, but they are present in different tissues of different biological species or classes or kingdom. For example, your Forsman proteins. These Forsman his proteins, they are found in the tissues of cats. They are found in the tissues of horse. They are also found in the tissues of pig. They are also found in the tissues of sheep, uh, uh, birds, plants. So these are uh, specific proteins which are present in different tissues of different classes or kingdom of life. So these are nothing but your heterogenetic or heterophile antigens. Now, coming to the next property, that is your size. Now, when you talk about the size, so uh, so substances with molecular weight less than 10,000 units are either weakly antigenic or non-antigenic. And very large molecules like not hemoglobin, hemocyanin, or are highly antigenic in nature. So the substances with low molecular weight are less antigenic, and the ones with larger molecular weight are considered as highly antigenic in nature. Now, talking about the chemical nature, so mostly proteins and polysaccharides are naturally occurring antigens, and proteins are more effective than polysaccharides. So, as I said, the antigens are basically proteinaceous in nature. So, these are either chemically they are proteins or polysaccharides, but the antigens which are proteins in nature are more effective when compared to your antigens which are polysaccharides in nature. However, gelatin, histones, and protamins are exceptions. Although these are proteins, they are non-antigenic. So there are certain exceptions in case of chemical nature, like though some are proteins in nature, but they are not considered as antigens. They are non-antigenic in nature. Now talking about the fourth property, that is susceptibility to tissue enzymes. So only those substances which can be metabolized and which can be converted into active form by the action of tissue enzymes acts as good antigens. So what are we saying? Here, the enzymes which can be metabolized at the same time, which can be converted into active form by the action of certain enzymes, specific enzymes, are considered as good antigens. And talking about your antigenic specificity, depends on antigen determinant site on the antigen. So as I said, antigen determinant site is nothing but your epitope. So antigen specificity depends upon your antigen determinant site or epitope. So antigenic determinant sites represents those active sites whose antibodies can react specifically. I mean, so what are we saying here? The antigen specific site is the site which bind to the antibody and can react specifically to that particular with that particular antigen. So antigen and antibody. So this is nothing but your the antigen specificity, and the remaining portion of antigen molecules is antigenically inert. So what are we saying? As I told you when we are discussing about the structure, the antigen has one or two functional epitopes. So only these two functional epitopes are responsible for binding with antibody and dealing with the antigen antibody reaction. So remaining part is considered as inert, antigenically inert. So this antigen specificity is again of different type, that is species specificity. So tissues of all individuals, in particularly species, possess, individuals having specific tissues, they possess specific antigen. Thus, human blood proteins and animal proteins are differentiated by antigen antibody reaction. So what are we saying here by species specificity means the uh, tissues are present in animals as well as in humans, but these tissues are different according to the different species. So even animals possess tissues and antibodies, antigens, whereas humans possess. But you cannot go and just take out the human blood and inject it into the animal blood, or animal blood cannot be injected into the. So these um, uh, there are variations between the tissues that are present in individuals of particular species. So that's why the human blood proteins and animal proteins can be differentiated by antigen antibody reactions. So in many cases, if at all there is any lost body found in the jungle or something, and if it has wounds, if you want to find out whether it has been done by any animal or by any human being, so we just go for forensic laboratory and we go for this antigen antibody. The blood samples would be taken and we undergo for antigen antibody reaction. Then we can come to a confirmation that this is done by humans or this is done by animals. So that is antigenic specificity. And that is nothing but your species specificity. Now, talking about your antibody structure. So, as we have already learned that antigens are nothing but proteins. So antibodies, when you talk about antibodies, antibodies are also defined as specialized serum proteins. These are also nothing but proteins. But how are they, from where are they produced? They are produced in response to antigenic stimulation. So generally, these antibodies are produced by one of the class of blood serum, that is nothing but your plasma cells. So B lymphocytes produce plasma cells and plasma cells produce antibodies. We have seen this uh, uh, I mean, sequence how they are produced when we are dealing with your humoral and cell-mediated immunity, how antibodies would be produced in response to an antigen. So antibodies are nothing but they are defined as specialized serum proteins, which are produced by one of the class of blood serum, that is plasma, in response to an antigenic stimulation. So antibodies generally react specifically with the antigen which stimulate their production. For example, a cold antigen has entered into your body, and immediately your body reacts to that antigen and the antibodies are produced. So this cold antigen will react with only that cold antibody. It will not react with the cough antibody. So it's not that cold and cough. I'm just giving you an example. So that is how the antigens and antibodies are specifically. Even if the cough antibodies are present in the and cough antigens are present and antibodies are present, they will not this if antigen enters into your body, the antibodies are produced in response to that particular antigen. Now this antibody will not say you stay back, I will go and take care of both the antigens. No. So the antibody that is produced or stimulated, that is produced in response to a particular antigen will bind to that particular antibody on to that antigen only. It will not go and bind to any other. So they are specifically, they have antigen antibody specificity. And chemically, antibodies are nothing but they are called as immunoglobulins. And these immunoglobulins denotes the chemical nature or chemical structure of a protein, while antibodies denotes biological activity and function of the protein. So when we say immunoglobulin, we represent the chemical nature. And when we say antibody, we represent the biological nature or the functions of the proteins. So that is how we generally use this terminology. So immunoglobulins are nothing but antibodies. But where we use this term immunoglobulins, when we are describing the chemical nature, when we are describing about the biological activity or the functions of the proteins, then we use the term antibodies. And see, generally, as I said, these immunoglobulins are synthesized by plasma cells and some tissues by lymphocytes also. So they're nothing but B lymphocytes. So talking about the structure of your antibody. So generally, when you talk about the structure of antibody, as I said, there are different types of antibodies: IgA, IgG, IgD, IgE, IgM. So basically, we take IgG as the extensively used antibody to describe the structure, and this IgG serves as a model of basic structure for all the immunoglobulins. So when you want to describe the structure of an antibody, we generally describe IgG structure. And it is a Y-shaped molecule made up of two identical heavy chains and two identical light polypeptide chains. And these two chains are held together by disulfide linkages or disulfide bonds. See, the long chains are called as heavy chains, which are represented as H, and the short chains are called as light chains, which are represented as L. And each light chain is attached to the heavy chain by a disulfide linkage. And two symmetrical head chains are held together by, again, disulfide bonds. So Porter is one of the scientists. Porter and his colleagues, they wanted to show the internal structure of the immunoglobulins. So what they did, they took an IgG and they split the IgG molecule by peptide digestion method, and they said that this IgG molecule have two identical Fab and one Fc fragments. Now, what are these Fab fragments and what are these Fc fragments? We will see in detail. So this is the structure of your antibody. So as I said, it has Fc region and Fab region. So this is Fab region and this is Fc region. It has heavy chains, it has light chains, and these are the disulfide linkages which bind these heavy chains with the light chains. And this is the site where antigen comes and binds with the antibody. So talking about the Fab fragment. That is, Fab is nothing but fragment antigen binding fragment. So there are two identical Fab fragments containing a single antigen binding site each. So each, as I said, Fab fragments are two, and these two will have one each of antigen binding site. And the portion of heavy construction, portion of heavy chain of Fab fragment is called as FD region. So as I said, you have heavy chains and light chains. So the heavy chains portion is called as FD region, and each Fab fragment contains a light chain and a part of heavy chain. So as I said, each Fab fragment has a heavy chain and a part of light chain. Now, talking about your Fc region, that is, Fc is nothing but fragment crystallizable fragment. region. So this Fc region lacks the ability to bind to the antigen. So that means this Fc region does not have anything to do with the antigen antibody reactions. Only Fab fragment is involved in binding with the antigen. So Fc region does not have any capability, or it lacks the ability to bind to the antigen. So each Fc fragment contains parts of both heavy chains, and it is the site for complement fixation, macrophagic fixation, skin fixation, regulation of catabolism. So that is where these Fc region comes into picture. So like in complement fixation, in skin fixation, in macrophagic fixation. So these fixation will be done in Fc region, but it will not be involved in binding the antigen to the antibody. So that is about your uh, structure of your antibody. Now let us see the different classes of antibodies. As we said, chemically, antibodies are nothing but immunoglobulins. So we have different classes of immunoglobulins, IgG, which is nothing but G represents gamma, and IgM, M represents macroglobulin, and IgA, A represents alpha, IgE, E represents epsilon, and IgD, D represents delta. So we'll see one by one what is in detail about these immunoglobulins. So when you talk about IgG, so this is one of the most abundantly available immunoglobulin in our body. So this serum globulin is the majorly available serum globulin, and it constitutes around 75% of the total immunoglobulins. And the molecular weight of this immunoglobulin is 150,000 daltons, and its half-life would be 2 days. So molecular weight, it is distributed equally between intra and extravascular compartments. And IgGs are the only immunoglobulins that passes through the placenta and provides natural passive immunity to your neonates. I told you when we were dealing with the humoral immunity that this is the only immunoglobulin that can cross placental barrier. None other than this can cross the placental barrier. So these IgGs can cross the placental barrier and produce immunoglobulin natural passive immunity to the neonates. So we call it as congenital immunity. And IgG appears later in infection but represents for longer period of time, and proteins from those microorganisms which are in blood and tissues. So these appear later. When immediately when antigen enters, these IgGs will not be seen. As I said, when the when the antigen enters into our body, immediately IgMs comes into the picture, and since they cannot handle the antigens for a longer period, they hand over the antigens to remaining IgGs like IgG, IgE, IgD, and IgA. And once the immunoglobulins IgG comes into picture, they comes at a later stage, but they last for a longer period. And IgG participate in most of the immunological reactions like complement fixation, precipitation, and neutralization of toxins and tissue viruses, etc. So when we are talking about the antigen antibody reactions, we can there are different types of antigen antibody reactions. So under this IgGs will participate in precipitate fixation and complement fixation, precipitate reactions, neutralization reactions, and all. Now talking about your IgM, that is macroglobulin, and this IgMs are one of the molecules of J chain, and it constitutes about 10% of the total immunoglobulins. So as I said, IgGs will constitute 75% of the total immunoglobulins, whereas IgNs constitute about 10% of the total immunoglobulins, and its molecular weight is 9 lakh daltons, and often called as millionaire molecule because it has so much of molecular weight. So 80% is confined to bloodstream and not found extravascularly, as these IgGs are found intra and extravascularly. So these IgMs are 80% it is confined to bloodstream and they are not found extravascularly. And this is the first IgG that, immunoglobulin that appears early following stimulation by an antigen, but persists for a short period. As I said, when an antigen enters into your body, the IgM would react first. So the first, in it, our body's immune system gets triggered and IgMs will be produced, and this will face the antigen initially. The first immunoglobulin to face the antigen would be IgM, but since it cannot handle the antigen for a longer period, it will hand over the antigens to remaining immunoglobulins. So this is considered as the first immunoglobulin that faces your antigen. And IgM cannot cross placenta. Its presence in serum of fetus or newborn indicates intrauterine infection. So if at all it is seen in the serum, or it is seen in the different fetus of the or in the newborn, then we can say the person is suffering from intrauterine infections, because other than that reason, these IgMs cannot cross the barrier and reach the newborns. And these IgMs are efficient in agglutination reactions, complement fixation, and cytolytic and cytotoxic reactions. So these are involved in different types of antigen antibody reactions. Now talking about your IgAs. So IgAs are nothing but alphas, immunoglobulin alpha. And second most abundant Ig, that is consisting of almost about 15% of your total human serum. So this is the second largest, mostly found immunoglobulin in your body. And IgA is found in mucosal surfaces and in secretions, which is called as secretory immunoglobulin. As I said, this IgA is considered as the first line antibody, because it is present in different secretions like mucous membrane, it is present in tears, it is present in lacrimal fluid, it is present in mucosa. So whatever the antigen it has to enter into the enters into the body, it has to enter through either uh, optical route or nasal route or oral route. So all these routes will have secretions, and in these secretions, the IgA will be present. So hence it is called as first line defense immunoglobulin. Its molecular weight is around 3,85,000 Daltons, and it is the dimer that is formed of two units joined together by glycoproteins, that is nothing but your J chains. And first globulin that is this immunoglobulin is highly concentrated in colostrum, saliva, tears, nasal fluid, sweat, milk, respiratory and genital and intestinal secretions. So as I said, this is the immunoglobulin that is present in different secretions. So this different secretions in which this Ig are mostly concentrated are colostrum, that is first feed of the mother, saliva, tears, nasal fluid, sweat, milk, respiratory, and genital and intestinal secretions. So these are the different secretions where you can find this IgAs, and they immediately attack your invading antigens. Now comes your IgE. So IgE is nothing but epsilon, also called as renin. So that is rain, REAGIN. And serum, here the molecular weight is 1,90,000 Daltons, and half-life is 2 to 3 days, and distributed extravascularly and produced by a very small portion of plasma cells living in intestine and respiratory tract. So there these IgEs are also present. These are specially involved in allergic reactions. So they produce histamines, and they try to prevent the allergic reactions in the human body. As I told you when we were dealing with the initial introduction, that these IgEs are, some people are allergic to dust or dust mites or house dust or pollen grains, and in chronic stage, this might turn into asthma or hay fever. But in that conditions, these IgEs will prevent the entry of these antigens. And it is unused IgE elevates the asthma, hay fever enzyme. Like, see, these are also one of the immune responses. So if at all, for example, if pollen grains, if the person is allergic to pollen grain, so what will happen once he smells some flowers and all, immediately he starts sneezing or coughing. So what is that? That is an immune response produced by the body. So these immune responses will be caused by your IgEs. Now talking about IgD, that is delta, Its molecular weight is 1,80,000 daltons, and it is present on the surface of B lymphocytes and serves as a recognition receptor for antigens. When we were dealing with humoral immunity, I told you that these are the mostly involved IGs that are present on the surface of B lymphocytes to recognize the antigens. So these IgDs are present on the surface of B lymphocytes and they help in recognizing your antibodies, antigens. So that is about your antibodies. So with this, we have completed the structure of antigen and antibody. You can find this information from textbook of pharmaceutical microbiology by NK Jain and also by JQB, and a lot of information is available in the net. If you have any doubts, just go through the topic once, and if you have any doubts, feel free to ask me anytime. Uh, I hope you understood the topic. With this, we have come to end of this topic. Let's meet with another new topic by in the next class. Thank you.